Good morning, everybody. My name is Sophie and I am a first-year genetics PhD student. This week I'm going to bring you into the lab with me. I'm going to talk you through why I am doing what I'm doing, but I'm also going to show you what I'm doing. So right now it is Monday morning. I'm heading in to check out my cells. I want to see how they're growing and how they grew over the weekend. I'm also going to look at some sequencing data, and based on that sequencing, I'm going to select some of my frozen glycerol stocks of E. coli to go forward with. Let's see how it goes. Okay, so my sequencing data was absolute trash, so I'm now redoing the overnight cultures and then doing a fresh mini prep tomorrow. I think my sequence data being so bad was something to do with my very low concentration DNA extraction rather than bad cloning. I'm not sure why the extraction was so bad, but it would explain why my sequencing data looks something like this. So right now I'm putting selective LB media into these 15 ml tubes. The media has ampicillin in it, and the plasmid that I've put into the bacteria gives them ampicillin resistance. So we call this selective media. It's going to select for the bacteria that have my plasmid of interest. So these are the colonies that grew up from my transformation of competent cells. So when I'm looking at the plate, I'm looking for one colony that's not too close to any others, so I can be very sure that I'm just picking up one pure colony. I'm then going to swizzle it around in that selective media and cap the tube up. So then I go to put them in the incubator at 37 degrees, 225 RPM overnight. That will give my bacteria plenty of time to grow up. Good afternoon, everybody. It's getting late in the day, but at least my cultures are on for overnight and my kit is ordered for tomorrow. So hopefully it goes better this time around. See you tomorrow. Good morning, everybody. Today we have a mini prep, and then hopefully I'm going to send them for sequencing. So the mini prep will give me the DNA, so the plasmid that I've been growing up in that bacteria, and then I'll send it away for sequencing to find out if it's correct. So that was a step that went wrong yesterday. I got my sequencing data back and it showed that my sequences were no good. It wasn't that the wrong bit was in or out or anything like that, but the quality was really low. And I think that's probably got something to do with how low concentration my extraction was. I think something's gone wrong, as that meant there's no real DNA in there, so the sequencing's failed. Still don't know why that happened, by the way, but we'll try it again this morning. Good afternoon, everybody. So I got my mini prep done as expected, and the plasmid concentrations were way better than last time. They were in that proper range of 100 to 200 nanograms per microliter for sequencing, so that's good. And I also got them sent off for sequencing, so we'll see how they look in a few days or so. So with that all done, it is time to go home and write everything up in my lab book. Normally I would write it up in my lab book immediately after I had done it, but I'm running a bit late in our booking system. So because of COVID, we book into slots in the lab and someone else is booked after me. So I've got to head home. Other than that, I'll see you guys tomorrow. Good morning, everybody. I hadn't really planned on coming in today, but my supervisor remembered that we want to revive some new cell lines. So with my gene editing, I want to show you that my experiment is reproducible, that anyone else with the same set of instructions could do it. So part of making it really robust and really reproducible is doing the experiment multiple different times and across different cell lines, that sort of thing. Also good news is that my supervisor looked at my fax data from my transformations, which I thought had completely failed, but it turns out I probably just analyzed them wrong. So I still don't have the actual efficiencies yet. I need to redo the analysis tomorrow, but it looks like I've made a mistake in analysis, not a mistake in the actual gene editing. So that's a big relief. Hello, everybody. All right, I am done. Can you tell from my hair that I wasn't intending to go in today? So today was very quick. I just ducked in, got those cells revived, and now they're happily growing in their little plates. I've also got a seminar this afternoon. My institute has like 2 or 3 seminars a week, so I will be attending that via Zoom. But I haven't actually looked at what the speakers are talking about yet. It can be a good idea to look up the speaker and have a quick read of some of their abstracts, so you've got an idea of what they'll be talking about. Just helps put it all in a bit of context. But that is also quite a nice, relaxing afternoon. I'll see you guys tomorrow for some fax analysis and fax reanalysis of my old data. See you then. This is the weather I was promised by Scotland. It's very fine rain, it's very cold, and I'm still getting very wet. The temperature and rain are slightly more bearable if I don't have my hand out filming, so I'll make it quick this morning. Today I am doing the third fax of my third transformations, but also I'll be re-looking at the data from my last fax to try and see where I went wrong with my analysis and what those results actually were. So I think I'll be in at work today for about 2 hours, and hopefully it won't be raining by the time I finish. See you then. Good afternoon, everybody. It is not raining anymore, which is fantastic, but it is still pretty cold. As my supervisor had said, my fact startup from last week was not all doom and gloom. The data actually seemed to show that my transfections that arced, that had that like spark in them, were less efficient, which makes sense. The ones that didn't arc had higher efficiency, so that lines up with what happened. So now that I've revived my new set of cells, I'm looking forward to doing transfections in those and seeing A, if I've improved my technique and get less arcs, and B, to see if I can get similar efficiencies with a new cell line. I'm also going to be sending the same lot of cells that I used for fax this week off for sequencing, so it'll be interesting to see if the sequencing agrees with the fax efficiencies. So I have generated a lot of data today, not just this week's fax, but also last week's fax reanalysis. So that's going to take quite a bit of writing up in my lab book to explain what I've done and why. So it's time to get home and start doing that. See you guys tomorrow. Good morning, everybody. I didn't get a chance to film on my way in this morning, but instead I will voice over this footage that I got today. So first things first, I'm suctioning out the media from this plate. That media is just basically going in the bin. The cells that I want are stuck down to the bottom of that plate, so I'm not impacting them by removing the medium. Then I'm going to trypsinize these cells, which basically means detach them from the base of the plate so that I can then spin them down and get just a pure cell culture. The goal today is to get these cells ready for DNA extractions, which I'll do next Monday, but I just want to get this first bit done. So this is my warm media. It's been warming at 37 degrees for at least 30 minutes at this point. It's going to neutralize the trypsin so that the cells aren't in any distress for too long once they have separated from the base. Seeing as I'm still waiting for the trypsinization to occur in the incubator, I'm just labeling the tubes that I'm going to need at a later step. Being able to think ahead in your long breaks is really valuable in speeding up your time. So in the background there, I'm checking out those cells under the microscope. I can see that they've trypsinized because they're floating in suspension instead of just sitting down on the base of the plate. Now I'm adding the nice warm media just to stop that process happening anymore. Now that my cells are in suspension, I can move them across to these 1.5 ml tubes, being very careful to use fresh pipette tips for each sample. So each of these wells represents a distinct gene editing event, so I want to make sure to keep them separate because the purpose of sequencing is to determine which was the best. So off camera, I spin these down and pop them in the freezer, ready for my extractions on Monday. Friday activities done. I have split down the cells that need it. I've got some DNA extractions ready to go for Monday, so they're sitting in the freezer waiting for me. So now my lab work is done for the week. Still got lab meeting, of course. I'm gonna go make a coffee and get some toast and get ready for lab meeting. And also there are a few points this week where I didn't have time to fill out my lab book as I went. I just took some small notes. So today I'm also going to spend some time moving those like dot point notes into proper lab book entries so that when I go to write this all up, I have all my notes ready to go. If you enjoy this content, don't forget to like and subscribe. I'm always interested to hear what you'd like to see more of. If you comment what you're interested in, I'll probably do more of it. See you guys next week.